Now we are going to talk about red blood cells, right, which are also called erythrocytes. Some of students know that red blood cells are called erythrocytes. That's the great news. So let's try to know something more than that. That red blood cells, first of all, they primarily are formed in the bone marrow. Actually, in prenatal development, hematopoiesis is in the yolk sac and then in the liver and spleen. So before the birth, red blood cells are first of all being formed in the yolk sac. Then as hematopoiesis shift to the liver and shift to the spleen, so red blood cells or erythropoiesis occur in the liver and spleen, and then it shift to the bone marrow. In a newborn, all the erythropoiesis is going on in bone marrow, right? So let's see in the bone marrow what are the different stages of development of red blood cells. Let's suppose here is your bone marrow house and here is your circulation. Now, in the bone marrow cells, the very first cell which is responsible for hemopoiesis is pluripotent stem cell, right? Pluripotent stem cell, and this divides into two types of cells. Yes, please. Lymphoid stem cell, and myeloid stem cell, right? Then myeloid stem cell can be divided, it can divide into, what type of cells? Three types of cells. Colony forming unit, erythroid, megakaryoid. Then colony forming unit, mainly into two. Colony forming unit, G, E, colony forming unit GM, granulocyte and monocyte, right? And from this erythroid and megakaryoid, right, it divides into two, right? Colony forming unit which goes in formation of megakaryopoiesis. Megakaryopoiesis means formation of platelets. And other cell is colony forming unit, colony forming unit erythroid. This is a happy cell we are going to derive RBCs, right? Again, this is a cell will concentrate, right, which is the progeny of this cell or derivative of this cell will eventually become red blood cells, right? But from where this cell is coming? Let's go back. The pluripotent stem cell divide into lymphoid stem cell and myeloid stem cell. Myeloid stem cell can give rise to colony forming unit erythroid megakaryoid cell, and this erythroid megakaryoid cell can make two colonies, that is colony forming unit erythroid and colony forming unit megakaryoid. And of course, colony forming unit erythroid will make the cells which will become red blood cells, and colony forming unit megakaryoid will eventually make platelets, right? We will concentrate on this, right? If we really concentrate on that, I need more space to explain it well. So let's make the bone marrow house bigger. Look, this colony forming unit erythroid, right? This is a large cell, about 20 micron. The first recognizable cell which is formed from here is proerythroblast. What is it? Proerythroblast. Now, proerythroblast goes into derivative cells and progressively cells get smaller. This is a very important concept, that when proerythroblast is differentiating into more mature cells, the derivative cells are progressively getting smaller, right? Let me tell you, this is which cells? Proerythroblast. Blast means rapidly proliferating cell. Proerythroblast. Then these three next stages are erythroblast. These three are, erythroblast. It means they are rather erythroblast. Next is also erythroblast, and then there is erythrocyte. cyte. Proerythroblast, then there is the erythroblast, and this erythroblast, which is the first one, this is called basophilic erythroblast because it takes blue stain, basophilic erythroblast. Then next takes some blue color and some red color. So we call it polychromatic. What we call it? Polychromatic. Right? Polychromatic erythroblast. And after that, this one is very much red. So this is called orthochromatic. Orthochromatic. Of course, this is also orthochromatic erythroblast. Rather make it erythroblast. Now, one thing which is important, that during the maturation of this process of erythropoiesis, the earliest cell is proerythroblast, which is going to, what is this? Basophilic erythroblast, then it goes to polychromatic erythroblast, then it go to orthochromatic erythroblast. Rather, because it does not multiply, so we call it orthochromatic normoblast. Orthochromatic please correct it, normocyte, because it does not multiply, sorry, normocyte. Now, let me tell you about their nuclear situation. What really happens, that in the proerythroblast, nucleus is very large, it has very, very fine chromatin, and it has prominent nucleoli. Right? And cytoplasm is basophilic, blue. It is basophilic. Now let me tell you why it is so. You know, chromatin is here fine chromatin. Fine chromatin means it is euchromatin. Euchromatin means loose chromatin. The chromatin in the nucleus may be condensed or it may be loose. Condensed chromatin is called heterochromatin and loose chromatin is called euchromatin. Actually, whenever cells are multiplying, they open their DNA and they are making replication of DNA. So whenever cell has to multiply or proliferate, it has to do replication of DNA, and whenever it has to do replication of DNA, then it has to open up its DNA and open up its chromatin. So chromatin will convert into which type? Euchromatin. Because these cells multiply a lot, erythroblast, right? So they have to have an open nucleus. Open nucleus means nucleus with loose chromatin, nucleus with euchromatin. Is that clear? Secondly, they are having very prominent nucleoli. You know, nucleoli are the factories to produce ribosomes. These are the places in the nucleus where ribosomes are assembled, and then ribosomes go to the cytoplasm. Because these cells in the future have to make a lot of proteins like hemoglobin, right? So for synthesis of protein, they need ribosomes. So because this cell has to multiply a lot, so it is keeping euchromatin, large nucleus, open nucleus with loose chromatin. And because it is going to make a lot of proteins like hemoglobin, so it has to synthesize a lot of protein synthesizing machinery like ribosomes. And ribosomes are assembled in nucleoli. So these cells have large nucleus, open nucleus with prominent nucleoli. Is that right? And because yet there is no hemoglobin, but some ribosomes are present over there and messenger RNA is present over there, so the cytoplasm takes blue color. Why the ribosomes take blue color? Because ribosomes have also RNA, ribosomal RNA, right? So DNA and RNA take blue color, they take basic dyes. Is that right? So whenever cytoplasm is rich in messenger RNA or rich in ribosome, cytoplasm will be Which colored? Blue colored. Right. Now what really happens? When erythroblast is stimulated, it proliferates and it gives rise to lot of basophilic erythroblast. Right? Basophilic erythroblast. Why we call this basophilic? Because here they have produced lot of ribosomes. And because they have lot of ribosomes, so cytoplasm become really intensely blue. Is that right? But nucleus become smaller. Nucleus become smaller. And it has euchromatin in the center, and it may have a little bit heterochromatin or condensed chromatin. Is that right? Now look, as early cells or precursor cells are going to more mature cells, right? The system should have three types of maturation. There are three steps of maturation. There should be size maturation, maturation in size. There should be maturation of nuclear material, nuclear maturation, and there should be cytoplasmic maturation. Cytoplasmic maturation. Listen. Size maturation means that as you are moving from early cells to more differentiated cells, in the process of erythropoiesis, size should become progressively smaller. That is size maturation. Nuclear maturation. Early cells are proliferating more, later cells are not proliferating at all, and eventually erythrocyte is without any nucleus. It means that early cells which are proliferating, they should have open nucleus. Open nucleus means nucleus with euchromatin. As you move forward, replication capacity is becoming less, proliferation capacity is becoming less. So open chromatin will go to condensed chromatin. So euchromatin will start converting into heterochromatin, right? And eventually, because the finished product RBC does not have any nucleus, it means nucleus should come out again. It means during the erythropoietic process, from precursor cells to the finished product RBCs, nucleus should progressively condense and then go out of the cell. Is that right? So initially nucleus is open, then some heterochromatin, then nucleus at this stage become more, what is this? Dense, more condensed, and here nucleus become very, very condensed, and it is about to leave. Is that right? So this process is called nuclear maturation. Third concept is cytoplasmic maturation. In the early cell, hemoglobin is not there. In the early cells, hemoglobin is yet not synthesized. But because cell intends to make a lot of hemoglobin, so it has to make a lot of enzymes and a lot of globin chains. So it means that this cell has a plan to make a lot of proteins. To make a lot of proteins, it has to synthesize a lot of ribosomes and messenger RNA. And these ribosomes and messenger RNA impart blue color to the cytoplasm. That is why early cells have basophilic cytoplasm or blue cytoplasm. But as cells are maturing their cytoplasm, gradually and progressively hemoglobin will appear in the cytoplasm. At this stage, hemoglobin appear. So some areas of the cytoplasm become hemoglobinized, and these areas are called, these look acidophilic or eosinophilic, and those areas where yet there is abundant, what is there? Ribosomes. And now at this stage in polychromatic cells, cytoplasm has blue shade as well as pink shade. Why? Because blue shade in those areas of the cytoplasm which are very rich in ribosomes and messenger RNA. You know, when one messenger RNA has a lot of ribosome, whole complex is called polyribosomes. So actually these blue colors are due to polyribosomes, and this red shade is coming due to presence of abundant hemoglobin. So this type of cell will give double shade. In some areas it will be stained bluish, in other areas cytoplasm will be stained red. You have seen checkers board. What are the colors used there? Black and white or black and red? Checkers or board? Okay, you can say this is a checker board with blue and red. Now So because it has double color, we call it polychromatic cells. What is this? Erythroblast, which is polychromatic. Precursor to this was not having hemoglobin, so it was purely blue, so it was basophilic. But when you go to the next stage, now basophilia is significantly reduced and cytoplasm is really too much mature, and it means too much mature means it is having a lot of hemoglobinization, and basophilia is far less, right? So when it is heavily hemoglobinized, right, we call it orthochromatic. What is the color called? Orthochromatic, right? So what we have learned during this process, that the first recognizable precursor cell of erythropoietic process is called proerythroblast. It's a large cell with an open nucleus, multiple nucleoli, right, and basophilic cytoplasm, right? It is about 20 micron. The next stage is usually 17 micron, then 14 micron, then 10 micron, and so forth. In the end, you have RBC, which is 7 to 8 micron. Progressively, size is reducing. So proerythroblast is large, open nucleus, multiple nucleoli, and basophilic cytoplasm. It multiplies into many basophilic erythroblast, and this basophilic erythroblast are a result of the mitotic divisions of proerythroblast, right? And these are having a nucleus which is smaller in size. Some heterochromatin appears. It means some chromatin is undergoing condensation now, right? And it means nuclear maturation has started, and cytoplasm even becomes more basophilic because this cell has now accumulated a lot of ribosomes and messenger RNA or polyribosomes. Am I clear? Then we go to the next stage, and the next stage is called, yes, polychromatic erythroblast. And this polychromatic erythroblast, why we call it polychromatic? Again, first you have to see maturation in size, maturation in nucleus, and maturation in cytoplasm. Maturation in size is now further reduced. We started with 20 micron, 17 micron, maybe this is just 13, 14 micron. Number 2, nuclear maturation. Now nucleus is more condensed, right, and more heterochromatin is there, and nucleoli of course disappear now because cell is no more synthesizing any more ribosomes. It had enough ribosomes, and there's evidence of fully formed, what is this? Hemoglobin, which is imparting which color? Red color. So blue and red shades, double shades are coming when you stain their cytoplasm, and these cells are called polychromatic, yes, erythroblast, right? After that, in the next stage, what really happens, that nucleus further mature and become very condensed. You see, it's very, very condensed nucleus, and it is about to be expelled from this cell. Is that right? Now, this cell, which is having a very, very mature nucleus, it has very heavy hemoglobinization. So due to that reason, basophilia become very less, and there is dominant eosinophilic cytoplasm. There is dominantly eosinophilic cytoplasm, a reddish color or pinkish color cytoplasm. Pinkish thing remind me something. I don't know, it's not relevant with the lecture, but it just remind me something. You see, a man went to see a very big boss of a CEO of a Fortune 500 company, right? And that man who was a visitor, he was stopped by the secretary. Secretary said that you cannot go in. My boss is talking to his girlfriend, and I cannot interrupt. So what happened? The visitor keep on sitting for 1 hour. Then he said, please check. The secretary again looked through the glass. She says, still you cannot talk to my boss. Now he's talking to his wife. Visitor got really very angry. He said, how did you know, sitting here just trying to fool me, you didn't talk to him, you don't telephone him, and fooling me that now he's talking to girlfriend, now talking to wife. How you know it? She says there are 2 ways. One reason is when he's talking to his girlfriend, he's holding the telephone like this. And when he's talking to wife, telephone goes like this. Right? And sometimes it goes like this. Second reason, when he's talking to his girlfriend, boss looks pinkish. Right? He must be orthochromatic boss. Pinkish. But when he is talking to his wife, he become bluish. Not only pinkish, he become bluish. Probably he cannot breathe well and cyanosis appears. And sometimes wife, if talk goes along with the wife, he walk in the office like sheepish. Like a sheep. Right? So she told the visitor that you cannot, under both circumstances, you cannot talk to the boss. Right? So as a good secretary knows by looking at the face of the boss, right, when he is pinkish and when it is bluish and what is the cause, a good doctor should know when he look at the bone marrow slide. Right? That if these are pinkish cells in erythropoiesis, a bluish cell in the erythropoiesis, what is really happening there? If you are having basophilic erythroblast, it is having more ribosomes, more messenger RNA and ribosomal RNA, and yet not having hemoglobin. But if you are having fully orthochromatic cell or highly eosinophilic precursor cell, right, then it is heavily hemoglobinized. It is towards more maturity. And of course, in between there is a stage of polychromasia. Now, I want to clear some confusion. These three cell stage have different nomenclature in different books. Some books say, every book called this cell proerythroblast. Every author call this proerythroblast. Thank God, somewhere they agree. After that, the confusion start. Let's try to manage that confusion. One way is to call all these cells normoblast. Early normoblast, okay, let me put it. Early normoblast, intermediate normoblast, and late normocyte. Right? Early mean basophilic. Intermediate mean double color. Right? Polychromatic. And late mean heavily hemoglobinized, reddish. Is that right? Of course, it is in early development and that is in late development. So this is one way to call it. All of them are suppose normoblast, early normoblast, intermediate normoblast, late normoblast, or preferably normocyte. Why this confusion? That in many books they write it as blast, but usually it does not undergo mitosis. That is why I write blast, because book write blast, but actually it is not blast because it does not proliferate. It is about to lose its nucleus, and anyone who loses the nucleus is not going to proliferate. Am I clear? So one nomenclature is early, intermediate and late normoblast, but late normoblast should be called normocyte. Clear? Number two, all of them can be called erythroblast, right? But basophilic erythroblast, then polychromatic erythroblast, then orthochromatic erythroblast. But actually it should not be blast because it does not multiply. Am I clear? But they call it blast. I don't know what to do with them. Erythroblast, but it is orthochromatic. Is that clear? Then we go to the next stage. Next stage, nucleus has gone out. And who will eat it up, this nucleus? Macrophages present in the bone marrow. They love to eat anything they find, including the nucleus of other cells. Right? So, what is left behind? Look at it. The left behind is a cell which is heavily hemoglobinized with a little blue network. A little blue network. Now, this type of cell is called cell with a network. Network is called retic. So this is a cell with a network, we call it reticulocyte. What we call it? Reticulocyte. And this reticulocyte is a fresh blood cells which appears in circulation. Reticulocyte is a fresh RBC which appear into circulation, but this reticulocyte is having some very little basophilia, which are the remnant ribosomes, right, with little messenger RNA and ribosomal RNA. This is reticulocyte. Within 1-2 days, with further maturation, what will happen? That its, of course, size will become a little more smaller and retic will disappear. It means ribosomes will disappear and hemoglobin formation will stop and cell will become purely hemoglobinized, and now it is called mature erythrocyte. Now it is called mature erythrocyte. What is it? Reticulocyte. Normally in our blood, reticulocytes are 1 to 2% of RBCs, right? If your bone marrow is working with the normal speed, if your bone marrow is producing erythropoiesis with normal way, then reticulocytes should be 1 to 2% of your, is that right? But if in your blood you find reticulocytes are more than suppose 10%, there is heavy reticulocytosis, it means bone marrow is underworking or overworking? Bone marrow is overworking, and there is fast what? There is fast erythropoietic process. So actually this reticulocytes percentage in the blood is very important for the doctors. That when laboratory gives a report reticulocytosis is suppose 2%, it is normal. But if you see in the report comes patient has reticulocytosis, reticulocytes with 20%, it means there is a very heavy reticulocytosis. It means in this person's blood there is fresh RBCs are coming from the bone marrow very rapidly, and there is accelerated erythropoiesis. Opposite to that, if you find someone blood, you take a sample and you study and there is no reticulocyte, it means the erythropoiesis has stopped. This type of problem occur in some aplastic anemias where immune system attack the stem cells, and if stem cells are destroyed, the fresh products are not made and there will be no reticulocytes in the blood. So in a way, reticulocytes are index of erythropoietic process. Is there any question? There is no question at all. So this was something about erythropoiesis. Now after this, we come to the characteristics of RBCs in the blood. Total erythropoietic process takes about 1 week, and this is under strong influence of erythropoietin. Right? Erythropoietin is a glycoprotein which comes from the kidney to the blood and then to the bone marrow. From the kidney, erythropoietin is released, erythropoietin. And erythropoietin goes to the bone marrow house, and it is responsible to stimulate the colony forming unit and produce more proerythroblast, and so that proerythroblast eventually convert into more RBCs. Is that right? So erythropoietin drives the erythropoietic process. But in the kidney, exactly in which area erythropoietin is produced? In the kidney, there are special tubes which are called, yes, nephron. Around these tubes, there are special capillaries. These capillaries are called peritubular capillaries. What are these capillaries called? Peritubular capillaries. Actually, it's the endothelial cells. Endothelial cells of peritubular capillaries have a capability to sense the oxygen in the blood. If partial pressure of oxygen in the blood is very low, and these endothelial cells are not getting enough oxygen, they start synthesizing and releasing erythropoietin, so that erythropoietin go to the bone marrow, right? And as erythropoiesis is accelerated and more RBCs come into blood, and oxygen carrying capacity of blood is increased. Oxygen carrying capacity of the blood is increased. Am I clear? Right. Now, after the erythropoiesis, now we come to some basic characteristics of RBCs. RBCs, basic characteristics of the RBCs. What is the size of an RBC? Who will tell me? Suppose this is a section of an RBC, right? The whole diameter, let's suppose, here it is 7 to 8, yes, you're right, micron meter, right? But at the edge, it is about 2.5 micron, and in the center, it is about 1 micron, right? And RBC mainly consists of membrane, cell membrane, and having some proteins which are associated with the membranes and lot of hemoglobin inside. Is that right? So first I will talk about membrane characteristics. As compared to the content of the RBC, RBC has extra membrane. Advantage of extra membrane is that it is biconcave disc. So surface area is more, so that hemoglobin can exchange the gases easily. Am I clear? Number 2, advantage of having extra surface area not only facilitate the gas exchange or oxygen uptake and release, it also facilitate the folding of the RBCs. When RBCs have to pass through very narrow capillaries, they are very flexible and they can fold on themselves and squeeze through narrow areas. Is that right? Now, let me talk about the membrane of the RBCs. Like all other cells, membrane of the RBC is lipid bilayer. It is lipid bilayer. But there is some very special characteristics of RBC's membrane, right? What is that? That there are certain proteins which are called integrin, these proteins are called integral proteins, which are through the membrane. And the other proteins which are within, what is this? Within the structure of, or under the surface of, interior surface of the RBC membrane, and these proteins are called peripheral proteins. These proteins make a network, and this network is held and attached with, what are these? Membrane proteins. Now this network of proteins which is present inside, you can say it's a network like this. What is the purpose of this network? Anyone who knows what is the purpose of this network of proteins? There is a network of proteins to the inner side of the RBC membranes, and this network is connected with the membrane through multiple proteins. Okay, let me tell you, let me give you an example that what is the real purpose of this. Listen, RBCs have extra membrane, is that right? So it is just like that a cell with less cytoplasm and more membrane. It is just like that if you are having small size, your own size is small, and you are having a dress which is very big, it reminds you something? Newly married ladies, you don't know bridal dress? You have never seen. At least you must have seen in movie, right? So females have very much extra dress, helping the textile companies. So now, actually RBCs are like those bridal, dresses having bridal dress, extra membrane. It does have an advantage. You can move the way you want. And is that right? So RBCs have flexibility and fold and gas exchange and many things, but they are having extra membrane. This is a disadvantage of extra membrane also, or extra dress. If you are having too much extra dress and you are passing through the narrow areas, what will happen? Your dress gets stuck. Let's imagine a fresh RBC, which is like a new bride having bridal dress with very big extra membrane or extra dress. Now, if a new bridal girl has to pass through narrow tunnels, passing through, and let's imagine that her dress gets caught up at multiple places, and wherever dress is caught up, she leaves behind a piece of the dress and moves forward. So progressively her dress will become larger or smaller? Smaller. When it will get progressively smaller, so what will happen? She will herself within the dress, as dress is reducing, she will remain elongated lady, or she will become circular, packed into a small dress? Packed into small dress. Is that right? Am I clear? Now, this is the risk in RBC. That RBC have lot of extra membrane, but it has to pass through very, very narrow areas of circulatory system. And if it does not have any special way to keep its membrane with itself, then when RBC is passing through the very narrow capillaries, it will keep on losing the chunks of the membrane. And eventually what will happen, that surface membrane for the RBC will become progressively less, and shape of the RBC will become spherical. Shape of the RBC will become spherical, because if volume is more and surface is less, automatically things become spherical. So RBCs have a risk of becoming spherical if they lose their membrane. And spherical RBCs, do you think they can bend and they're flexible? No. So nature prevents this type of tragedy from RBC. How? The nature has provided this network of proteins which hold the membrane of the RBC tightly with the substance of the RBCs. It's just like that, a bride which is having extra dress, but inside there are too much elastics which are keeping the dress with her. As she keeps on moving, if dress is stuck, with elastic it is pulled. So she does not lose her dress at multiple places. Am I clear? So these are the membranes, these are the special protein networks, these are called structural protein of RBCs, right? These structural proteins have a very important function. They keep the membrane of the RBC highly flexible and stable, and they keep the membrane of the RBC with the substance of the RBCs, and these structural proteins don't allow the RBC membrane to be lost here and there when RBCs are passing through narrow capillary structures. Am I clear? No problem up to this? Now, what is the importance to learn it? If you really want to know the names of these proteins are called spectrins. Spectrins, right? These black proteins are enkyrins, right? These proteins are called glycophorins, or some of them are called 4.1 band proteins 4.1, right? There's no need to remember all of these names. If you really want to remember the name, remember just two, spectrins and enkyrins. But there are many proteins which are involved. There are spectrins, and there are actins, and there are tropomyosins. There is band 4.1, there's band 3 protein, there's glycophorins. No need to remember, just remember the basic concept that, look here, this is the membrane of RBC, and there's a network of proteins under it, spectrin, and ankyrin and other protein anchor this network with the membrane. So that when membrane is getting flexible, these proteins keep the membrane and don't allow the membrane to be pieces of membranes to be lost. Am I clear? Now, let's look at the clinical evidence. If in erythroblast, if for example, the genes which synthesize these structural proteins, if those genes are mutant and this network is not stable, either they are deficient spectrin or there is deficient ankyrin. Is that right? Again listen, if you inherit defective gene for ankyrin or spectrin or any other component, can you make a good network? No. This RBC, which is produced by a person who has mutation in these protein structure or mutation in the genes which make these proteins, such RBCs with mutant network or weak network, when they will come into circulation, when such RBC will pass through narrow capillaries, can it hold its membrane tightly? No. So this RBC will lose its membrane here and there, and eventually these RBCs become spherical. These RBCs will become spherical, and the disease will be called hereditary spherocytosis. Because when you take the sample of the blood, you find RBCs are spherical, not biconcave. Let me tell you how you decide on the slide that RBC is biconcave or spherical. Look, in biconcave, there is more hemoglobin on the edge and less hemoglobin in the center. So in healthy biconcave, there is more pinkish area on the side and central area is pale. If you find an RBC under the microscope, center is pale and periphery is pink, it means this is normal biconcave RBC. Are you understanding? Because central should be pale, it has less hemoglobin, and biconcave edges are having more hemoglobin. But when it is spherical, when it is truly spherical RBC, then there is more hemoglobin in the center, isn't it? So this RBC will stain without any central pallor. So when you find an RBC under the microscope without any central pallor, it means this is not biconcave, it is spherical RBC. So if you, in these patients, because spectrin or ankyrin or related proteins are mutant, and these RBCs are not having the proper capability to keep the membrane with the RBC, so such RBCs during the circulation keep on losing the membrane and they become spherical, and this condition, because it is inherited, so we call it hereditary spherocytosis. Hereditary spherocytosis. But if this problem is mild, then we call it hereditary elliptocytosis. If it is severe, then hereditary spherocytosis. If problem is mild, then RBC become elliptical, not fully spherical. Then we call it hereditary elliptocytosis. Cytosis. Am I clear? So now you will remember what is the purpose of these proteins. This is a very important structural protein related with the RBCs. And of course, what happened to these RBCs? When RBCs become elliptical or spherical, can they bend? No. Do you think it's a very fat man passing through narrow tunnel? Can he fold on himself? No. So such RBCs get stuck into narrow areas, especially in circulation of spleen. You know, in the spleen there is special type of circulatory area through which RBCs have to squeeze their way, and if a elliptocyte or spherocyte happen to be there, it will get stuck there, and then who will eat that RBC? Macrophages. So such RBCs are very rapidly eliminated from circulation. So we say there is hemolysis, and due to hemolysis their RBC count and hemoglobin concentration become less, and we say hereditary spherocytosis is a form of hemolytic anemia, because hemolysis occur and RBC count and hemoglobin concentration goes down, so there is hemolytic anemia. Am I clear? There is no problem about this. So this was something about their structural protein. Then there are enzymes also within the RBCs, but remember RBCs don't have mitochondria, so they break down the glucose only partially. They do only glycolysis, so they have enzymes related with the glycolysis. Or RBCs can make energy through hexose monophosphate shunt. So they do have enzymes for that system as well. Is that right? So they have structural proteins and functional proteins. Structural proteins are spectrins, ankyrin and related proteins which are responsible to keep the cell in biconcave shape and responsible to stabilize the RBC membrane. And then the functional protein and functional proteins are enzymes related with RBC metabolism. Is that right? And of course RBC does not have any nucleus. Is that right? And usually its life is how much? 120 days. Around that time their proteins become denatured. They don't function well. So RBCs become somewhat, the membrane becomes somewhat rigid, not as flexible as they are. As newly born RBCs are highly flexible. As RBCs become older and they become more senior citizens, then they are not so flexible. They cannot twist through certain situations, and they get stuck into capillaries of spleen, and their macrophages will eat them up. Am I clear? Right? Then a very important protein, the most important protein which is present in the RBC is hemoglobin. So let's talk about hemoglobin. But I will talk very briefly about it, that basic structure of hemoglobin and types of hemoglobin. Actually, during the synthesis of hemoglobin, the first molecule which is made, this molecule is called pyrrole ring. So erythroblast first of all produce pyrrole rings. Then 4 pyrrole rings are put together, assembled. These are 4 pyrrole rings. If they are put together, this structure is called protoporphyrin. Then, if this is protoporphyrin, in the center of protoporphyrin, if you put iron, what you put? Iron. Then it is called heme. Is that right? Again, erythroblast make initially pyrrole rings, assemble them into protoporphyrin rings, and protoporphyrin ring, in the center of that they put iron, and then there is heme. And then they synthesize the globin chain, and with the heme they put one, this is heme, protoporphyrin ring with iron, and they put a globin chain, right? And now what is this called? Hemoglobin monomer. What is this called? Hemoglobin monomer. But basically hemoglobin is a tetrameric protein, right? So there are tetramers. Now listen. Actually, there are different type of globin chains which can be synthesized. This was suppose alpha globin chain. Erythroblast can also produce beta globin chain, which is called beta chains, and it can also produce, yes, gamma globin chain, and it can also produce, yes, another type of, all of them of chains are with the heme, and this chain is delta chain. So globin chains can be alpha chains, beta chain, gamma chain and delta chain. Is that right? Now, these are different type of monomers of hemoglobin. Now, if there are two monomers with alpha chain plus two monomers with beta chains, then this hemoglobin is called hemoglobin A. A for adult, because in adult most of the hemoglobin has two alpha chains and two beta chains. Then in adult another variety is found, 96% of the hemoglobin is of this configuration. About 2 to 3% hemoglobin is two alpha chains with plus two delta chains. That is called hemoglobin A2. That is called hemoglobin A2. Then in fetus, usually two alpha chains are combined with two gamma chains, and that is called fetal hemoglobin, hemoglobin F. Is that right? So what we really understand that alpha chains are present in all hemoglobin, adult hemoglobin or A2 hemoglobin or F hemoglobin. But actually, if two alpha are with two beta, this will become adult hemoglobin A. If two alpha are with two delta, then these are hemoglobin. And if there are two alpha with two gamma, then it is hemoglobin F. Now let's have a test. If two alpha with two delta, what will they make? Hemoglobin A2. If two alpha with two beta, hemoglobin A. If two alpha with two gamma, hemoglobin F. Another important thing. Actually, fetal hemoglobin with gamma chains, it has more strongly, it has more strong affinity for oxygen, so that it can really pull the oxygen from maternal circulation to fetal circulation. So fetus is very clever. It does not activate beta chain or delta chain genes, it activates gamma chain genes. So in the fetus, hemoglobin is 2 alpha 2 gamma. Actually, why this hemoglobin has an advantage for the fetus? Because this hemoglobin has stronger affinity for what? Oxygen. So it can pull the oxygen from the circulation of the mother to the fetal circulation. Is that right? Okay. Another thing which I would like to mention here, that if there is some mutation in the genes which produce these chains, right, then there can be problems. For example, if a person has mutation in mutant beta chains, so he is not making the beta chains at all. Let's suppose these are the genes. This is alpha gene, this is for beta gene and beta gene, this is gamma gene and gamma gene. I'm just giving you an example. Let's suppose if beta genes are defective, then what will happen? This person initially in fetal life will make alpha with gamma. So that is which gene? Which hemoglobin? F. But actually this baby after the birth, he should make alpha with beta. But if beta are defective, he will keep on producing alpha with gamma. Is that right? This type of diseases in which there is quantitative deficiency of any type, some type of globin chain, this type of diseases are called thalassemias. Have you heard of thalassemias? Thalassemias are diseases in which there is quantitative problems with the production of globin chains, right? And certain globin chains are not produced with proper ratio, right? Thalassemias. Right. Then we come to another concept. Do you know something about blood groupings, ABO and RH blood grouping? You want to know something or you know it well? You want to know it?
Yes.
Okay, I will talk very briefly about that. That there are so many ways to group the blood group, right? Typing of the blood grouping can be done, but two important grouping I will talk about is ABO blood grouping and RH blood grouping, right? First, let me tell you what is meant by the ABO blood grouping. Actually, on RBC surface, right, different people express different antigens, right? Some people express, okay, I will make a full chart here. Some people on the RBC surface, they only express A antigen, and some people express only B antigens. Let's suppose these are 4 different persons. Person number 1, person number 2, person number 3, person number 4. Now, person number 1 on his RBCs, he is expressing only A antigen. Is that right? A antigens. The persons who express A antigens on their RBCs, they are called blood group A. It's so easy. And people who express B antigens on their RBC surface, they are called blood group B. Some people are expressing the both antigens, right? So they are called blood group AB, right? AB. And some of them don't express, they simply express an H antigen. Actually, H can be modified into A and B. H antigen is expressed by all of them. H can be modified into A or can be modified into B. And some people H is there and it is not modified into A and B. And H is not immunogenic, it does not stimulate the immune system. So practically we can say these people do not have A and they do not have B. Are you really clear what I'm talking about? No? Yeah. Right. So H antigen is present, H molecule is present on all of them. It can be modified into A or can be modified into B. And in some people, the enzymes which have a capability to modify into A and B, they are not there. So practically they don't have A, they don't have B antigen. So we call them blood group O. So in a way, this is blood group A, right? This is blood group B, this is blood group AB, and this patient has O. O is no A, no B. Is that right? Now there is another thing. Actually, A and B antigens are present in environment also, and these antigens are also present in microbes. So when we eat, many of these A and B antigens come to our GIT, and these antigens which are coming through foreign sources, they try to stimulate our immune system. They try to stimulate our immune system. Now listen. For example, person number 1 is blood group A. Now in blood group A person, A and B antigen will be coming to his immune system stimulation from the microbe and from the diet. But when A antigen will come, A antigen will be perceived as self antigen because this antigen is coming from environment, but it is also present in his own body. So A antigen, because it is his own antigen, do you think his person's immune system will make anti-A antibody? No. So in this person, the person who has A antigen, he does not make anti-A antibodies. But when B antigen come to his system, right, and B antigen is not present in his own system, so his immune system is going to perceive B as foreign antigen, so he will make anti-B. So what really happens, that antibodies present in blood group A patient are anti-B. And people who are blood group B, right, when their immune system is challenged by A and B foreign antigens, they will not make anti-B because B is their self, but they will make anti-A. They will make anti-A antibodies. These antibodies are called also hemoglobins, right? Anyway, person who has A and B both antigens are self antigens, when foreign A and B antigens come through microbe and diet, do you think their immune system will make anti-A or anti-B antigens? No. So they don't have antibodies. But person who has no A antigen, no B antigen, whereas in this person's body, through the diet or through the microbe, A and B antigens come, he is going to make anti-A antibodies as well as he is going to make anti-B antibodies. So is it clear? That again, person who has blood group A, he is having A antigen on the RBCs and in his plasma he has anti-B antibodies. Person who has blood group B, he has which antigen on RBCs? No. Which antigen on RBCs? B. And which antibody in the blood? Anti-A antibody. Right? Person who has a blood group A and B, he is having which antigens expressed on RBCs? A and B both antigens. And in his plasma which antibodies are there? No, there are no anti-A, no anti-B. Other antibodies against other antigen may be there in the body. You make him totally immuno-incompetent. Okay. Person who has blood group O. Yeah. Person who has blood group O, he does not have A antigen, he does not have B antigen. So he will develop anti-A and anti-B. Is that clear? Now, we talk about ABO incompatibility. You heard of this, isn't it? Now we talk about ABO incompatibility. First, listen a basic rule. Let's suppose you are going to do a blood transfusion. This is the circulatory system of recipient person, right? If you bring the donor's RBC, these are the donor RBC which are going in his body, and this is the donor plasma which is also going to his body. Is that right? Now listen carefully. In donor RBC, let's suppose this is donor RBC is A. It means donor blood group is A. And by mistake, if you give A blood to a person who is B group, it means this person has which antigen on his RBCs? B. Recipient has a blood group B, and unfortunately you have given the recipient a blood which is blood group A. So recipient RBCs have B antigen and anti-A antibodies, anti-A antibodies, which are already present in the serum of or plasma of recipient. And when you bring the donor RBCs, which are having concentrated A antigens on the RBC membrane, anti-A will attack the donor RBCs and destroy the donor RBCs. So what really happens in ABO incompatibility, these are the donor RBCs which have a risk of being destroyed in recipient circulation. I'm the recipient. Okay, let's suppose this is the donor. Please come here. Let's have a look on the donor. Right? Come on this side. Let's suppose this person has blood group A and I'm having a blood group B. His RBCs are having which antigen? A antigen. My RBCs are having B antigen. His plasma has which antibody? Anti-B. And my plasma has anti-A. Clear? Now, if by mistake you put his blood in my circulation, when his RBCs will come to my circulation, his RBC membrane has thousands and thousands and thousands of A antigens concentrated. And my plasma has anti-A. So my anti-A will attack the donated RBCs and destroy them. And hemoglobin will be released in my circulation from the ruptured donor's RBCs. Hemoglobin is released and this free hemoglobin will damage my kidney, right, produce kidney failure, activate lot of mast cells which will release lot of histamine, and my blood vessels will dilate, I may go into shock. That is a dangerous situation, right? So donor's RBCs in case of ABO incompatibility will be destroyed in recipient circulation and produce problems. But another important point. But donor's antibodies, he was having anti-B antibodies. His antibodies will also come to me, but they will get rapidly diluted in my circulation, right? So donor's antibodies usually do not attack recipient RBC, but recipient's antibodies do attack the donor's RBCs if there is ABO incompatibility. Am I clear to everyone?
Why don't his antibodies attack your antibodies?
Look, his antibodies are in plasma. He is having anti-B antibodies. Now, his plasma fluid is diluted into all my plasma, right? And antibodies become so subcritical concentration that they don't effectively destroy my RBCs. Is that right? But when his RBC comes, his RBCs are having thousands and thousands of antigens concentrated on his RBC membrane. Can those antigens get diluted in my body? No, they remain concentrated on donor's RBC surface. So they are very effectively attacked by my antibodies. So what really happens in ABO incompatibilities, these are the donor RBCs which are attacked by the recipient antibodies, and donor's RBCs rupture in recipient's circulation and produce problems. But donor's antibodies get diluted in the huge volume of plasma of the recipient person. Am I clear to everyone? Please have a seat. You are about to kill me.
Good job, Hans. So,
Now, yeah.
so it's the hemoglobin from the donor's RBCs that causes all the problems?
Yeah, hemoglobin, which is actually when donor's RBCs rupture in recipient circulation, then free hemoglobin produces the problem. Is that right? For example, free hemoglobin can leak through glomerular membranes and damage our kidneys, especially proximal convoluted tubules. Anyway, let's come back. So now we talk about it. The person who has blood group A, can you give him A blood group? Yes. Can you give him B blood group? No, because he has anti-A. B blood group we cannot give, because when B RBCs will enter into A circulation, A person has anti-B antibodies, so donated RBCs will be ruptured. Can he be given AB blood group? No, because again donors RBCs will be destroyed by anti-B. Can he be given O?
Yes.
Yes, because O does not have A or B, so anti-B cannot destroy the O-coated RBCs. Clear? Now we come to person who has a blood group B. He has which antibody? Anti-A. Can he get blood group A? No. Can he get blood group B? Yes. Can he get AB? No, because anti-A will destroy the A RBCs, A antigen. And can he get O? Yes. Now we come to the person with blood group AB. So this person does not have any antibody, no anti-A, no anti-B. Can he receive blood group A?
Yes.
Can he receive blood group B?
Yes.
Can he receive AB?
Yes.
And can he receive O? Yes. Why? Again, person who has blood group AB, he does not have anti-A antibodies, he does not have in circulation anti-B antibodies, so he receives A RBCs with A antigen or B antigen with AB antigen or O antigen. None of these RBCs are destroyed because he is not having anti-A or anti-B antibodies. But the person who has blood group O, he is having anti-A and anti-B antibodies. So can he receive, should he receive the blood group A? No. Should he receive B? No. Should he receive AB? No. Should he receive O? Yes. Now if you really look at it, what we see that every group can receive the O. Is that right? Because O RBCs of O blood group do not have A and B antigen. So whatever person's circulation they go, they will not be destroyed by anti-A or anti-B. So blood group O people are said to be universal donors. They are said to be universal donor. Opposite to that, when we talk about AB blood group, right, they can receive blood from everyone, because people who have AB blood group, they do not have anti-A antibodies, they do not have anti-B antibodies, because they do not have anti-A antibodies and anti-B antibodies. So you put A, B or AB or O RBCs, they will not be destroyed. So they are universal recipient. So ABO blood group is universal recipient and O blood group is universal donor. Is that right? I hope this concept is clear. Right. Now we talk about RH blood group. Only few words. Or you want to have a break? Now, RH antigen is present in some people's RBCs and it is not present in some other people's RBCs. RH antigen was first of all discovered in some monkeys, rhesus monkeys RBCs. So that is called rhesus antigen or RH antigen. Now, the RBCs which are having RH antigen, right, RBCs which have RH antigen, they are said to be RH positive, and RBCs which do not have RH antigen, they are said to be RH negative. And if I say my blood group is A positive, it means I have A antigen plus RH antigen. If I say your blood group is A negative, it means you have A antigen but no RH. If I say you are AB positive, it means you have A antigen on RBCs, B antigen on RBCs as well as RH. If I say you are O negative, it means you do not have A antigen, you do not have B antigen, you do not have RH. You understand this concept? Now, the special precaution which we have to make about RH system is, we'll talk about that precaution later. First, as I told you, A and B antigens are present in nature. So they come to us from microbes and from the diet. RH antigen is not present in nature abundantly, not present in microbes, and RH antigen is not present in diet. So it is not normally coming to us. Is that right? Now, if I am RH positive person, my RBC is a RH positive person, right? Because from the nature, no Rh antigens come, I will not make anti-Rh. But even if you put some other person's RBCs which are Rh positive to my circulation, still I will not make anti-Rh because Rh antigen is considered my self antigen. Am I clear? Now, but if there is a person who is Rh negative, if there is a person who is Rh negative, in that person normally anti-Rh antibodies are not present. Are you understanding? Person who are Rh negative, they don't make anti-Rh antibodies spontaneously. But a person who is Rh negative, if that person get Rh positive RBCs in her body, then that person can make anti-Rh antibodies. Am I clear? Now I will give you a classical example which is also clinically related. Let's suppose there is a woman who is Rh negative. You know, some women are Rh positive and some women are Rh negative. Let's suppose there is a woman who is Rh negative and she gets pregnant and unfortunately she is carrying a baby which is Rh positive. Of course, that Rh positive gene came from paternal side, right? Let's suppose, look at her situation. This is maternal circulation, mother circulation. Her RBCs are Rh negative. She does not have Rh antigen. And here is the happy baby, right? And this baby has an RBC which is Rh positive. This, she is first time pregnant, and this is Rh negative mother which is carrying Rh positive baby. Now what really happens that during the pregnancy, but mostly just after the delivery, a little blood from the baby goes to the mother, about half ml blood, right? So what really happens when this is the first baby. When first baby is delivered, when this woman which is Rh negative, she was carrying Rh positive baby, fetus, and when she delivers first baby, at the time of delivery, when placenta detaches from the uterus, a small amount of fetal blood goes to the mother. And then mother, this baby is out of course. Right? This first baby is out, but during its delivery, some of its blood went to mother, which was RBCs which are Rh positive. No, fetal RBCs were Rh positive. This baby has gone out, no trouble to it. But when mother immune system will receive Rh positive RBCs, do you think Rh antigen is natural for mother? No. So mother will make anti-Rh antibodies. Now this lady who is herself Rh negative, but she is carrying which antibodies in her blood? Anti-Rh antibodies. She will not have any problem herself because her RBCs are Rh negative, and anti-Rh antibodies she has made due to stimulation coming from the first baby. These antibodies will not disturb her. Let's suppose after 2 years she again becomes pregnant. Now she is having the second baby, right? And this baby is also Rh positive. This baby is also Rh positive, right? But now mother circulation is different. You know, the naughty first baby, it stimulated mother immune system on the way when he was out, and mother made anti-Rh antibodies. So mother was having anti-Rh antibodies due to her prior exposure to Rh antigen. So when she will be pregnant and carrying the second fetus, anti-Rh antibody belong to IgG group and it can cross placenta. So it will come to this baby, and this anti-Rh antibodies from maternal plasma come to the fetus and bind with the fetal RBCs and destroy them. So when this baby will be born, this baby will have anemia, severe anemia, because hemolysis is going on, because maternal anti-Rh antibodies are destroying baby's RBCs. But this second baby also gives some, at time of delivery, gives some Rh positive RBCs, and mother immune system now make more anti-Rh, and trouble is more for the third baby. You know, life is strange. This time she is carrying the third baby. I think this must be right? Why? Because first baby stimulated the mother by giving a little blood and mother made anti-RH antibody. Second baby also gave RH positive RBCs to the mother. Mother made a lot of anti-RH antibodies. And lot of anti-RH antibodies come, and now baby's RBCs are destroyed in very seriously. And when the severe destruction of RBCs, then to compensate for these RBCs loss, baby has excessive hematopoiesis. And due to excessive hematopoiesis, baby's liver and spleen enlarge. Because in the fetus, liver and spleen act as hematopoietic organs. But by the time of birth, most of the hematopoiesis is from the bone marrow. But because in this baby, a lot of destruction of RBCs are going on, so liver and spleen continue their hematopoiesis and they become enlarged due to increased hematopoiesis. And bone marrow also does. So this baby is born with hepatomegaly and splenomegaly and still a lot of what? Severe anemia. And a lot of these RBCs which are destroyed, they are releasing bilirubin, unconjugated bilirubin, which may go to the brain of this baby, because babies are having, newborn are having immature blood-brain barrier. So when these RBCs are rapidly destroyed, they release the they release a lot of hemoglobin, which is eventually releasing bilirubin. Breakdown product of RBCs is bilirubin, and this heavy amount of bilirubin may cross the blood-brain barrier, and this may go to the baby's central nervous system, destroys cortex and basal ganglia. So this baby will develop really weeping much. This baby will develop mental retardation and motor disorders and problems like this, right? Woman is determined to have more babies.
Don't they give them medicine after the first pregnancy?
Yeah, they will. There will be some good doctors like Dr. Gigi will be there and she will do some treatment. Yes, we'll talk about this for sure. Now, unfortunately, she was in the third world, not taken care properly, and she is very keen to have more children. Now this time what will happen? The real sad thing. Again the baby is RH positive. And what will be the problem with this baby? The mother has so much RH anti-RH antibodies that very severe destruction of baby's RBCs. And baby has a very large liver, very large spleen, and still very heavy hematopoiesis cannot match the losses of RBCs. So blood becomes very thick or thin. Thin, because most of the RBCs are destroyed. And heart of the baby fails to maintain the circulation. Is that right? So baby also develop cardiac failure. And in advanced classes you will know, when someone develops cardiac failure, there is generalized edema. So this baby has hepatomegaly, splenomegaly, and due to heart failure, baby develops generalized edema. And this baby will be very much edematous baby. So we call it hydrops fetalis, fetus full of water, hydrops fetalis. And mother cannot maybe deliver it through normal birth canal. During this process, either mother will die or some doctor in the third world, he will cut the baby into pieces and bring it out to save the mother life. Or you will do C-section if the facilities are available and bring the baby out, but it's full of water. Is that right? And usually many of these babies die within the uterus, right? Mother is already having one child which is mentally retarded, and now she has this tragedy. Right? Another problem during this situation occurs that because in these babies hematopoiesis continues in liver and spleen. Right? Now, hematopoietic function in liver and spleen is different than hematopoiesis in bone marrow. Bone marrow is such a clever organ that it only allows mature product to go into circulation. You are understanding? From bone marrow, when bone marrow is doing hematopoiesis, do you think pro-erythroblasts come into circulation? No, only reticulocyte and RBCs come into circulation, right? So actually, your bone marrow stroma is so much, you can say, so well functional that it only allows the mature RBCs, WBCs and platelets to go into circulation. But liver and spleen are not so good hematopoietic organs. And if they continue their hematopoiesis for a long time, they allow the precursor cells also to jump into circulation. So in these babies who are born with continued hematopoiesis in liver and spleen, they have a lot of erythroblasts into their circulation of these babies. When these babies were born, right? Doctors took their cord blood and tried to see what is wrong, and they found that these fetuses which were having severe jaundice or having hepatosplenomegaly, or in severe cases having, what is that? Generalized edema, they found in the blood of these babies or newborn fetus there are a lot of erythroblast. Because what is the underlying cause for that erythroblastosis present in the blood? Because hematopoiesis are going in liver and spleen, and they allow the immature precursors also to come into blood. So doctors at that time, when they never understood the mechanism of this disease, they just said this is a disease of a fetus in which lot of erythroblasts are found in the blood. So they called this disease erythroblastosis fetalis. What was this disease called? Condition of the baby? Erythroblastosis fetalis. Am I clear? So that is why now, yes, we come to Dr. Gigi. The Dr. Gigi was asking, is there any treatment? She is very much concerned. So we should be concerned about patients. There is treatment now available. What we do, that now commercially available anti-RH antibodies are there. There are injections available, injectable forms of anti-RH antibodies. The injection comes with the name of Rogam. Right? Now, RH immunoglobulins, Rogam. Rogam, the injections are available. What doctors do, it's international standard of pediatric, you can say, obstetric care, that any woman who is RH negative, right, as soon as she gives birth to a baby, immediately baby's blood should be checked. If baby is RH positive, immediately mother should be injected, within 72 hours, mother should be injected with injection of Rogam. What is the advantage of injecting the mother with Rogam? Let's suppose this was her first baby born. We already knew mother was A negative and baby is supposed AB positive. So it means RH negative mother has given birth to RH positive baby. So probably some babies' RBCs are present in maternal circulation, and these RBCs are RH positive. We inject the mother with anti-RH antibodies. These antibodies will come, and these antibodies will bind with the circulating fetal RBCs and destroy them. When these fetal RBCs are rapidly destroyed in mother circulation, so these RBCs are never detected by the immune system, so immune system never make anti-RH antibodies. In this way, mother becomes safe for the next baby. So this is international practice now, that whenever RH negative mother give birth to RH positive baby, mother within 72 hours of the delivery of the baby should be injected with Rogam. Right? These are anti-RH antibodies. We also call it anti-D antibodies. RH antibodies, RH gene or RH antigen has 3 types, D, C and E. Right? Anyway, these antibodies are also called Rogam antibodies or anti-RH antibodies or anti-D antibodies, so that any mother who is RH negative herself give birth to RH positive baby, that mother should be injected with anti-RH antibodies, so that in fetal RBCs in maternal circulation should be coated by those antibodies. And antibody-coated RBCs are rapidly eliminated by the macrophages of the mother in spleen. So maternal circulation becomes free of fetal RBCs. And because RBCs are rapidly destroyed, so if they are present, fetal RBCs are present for a very short time in maternal circulation, they don't produce enough stimulation to the mother immune system, so mother does not produce enough anti-RH antibodies. Yes, what's your question?
Is IgG able to cross the placental barrier, or is it IgM?
Pardon?
Is IgG able to cross the placental
IG?
barrier?
G. IgG, yes, this is IgG antibody crosses the placental barrier. This has some benefit also, that IgG antibodies, for example, a woman who is from Africa, she has been exposed to African infections. So all her life he kept on making anti-IgG antibodies against many microbes. When she will have a baby, so all the IgG antibodies she has in her circulation, they will go to the baby, and baby will be protected against those infections. Is that right? So every woman who becomes pregnant, whatever pool of IgG antibodies she has, she gives it to the baby. Is that right? So that when baby is born to the new world, right, it should have some degree of protection, immunity. Am I clear? Any more question? Yes.
What if the
Now another thing, why IgM does not cross? Listen, when anyone get infection, initially you make IgM, but if infection come again and again, you make IgG. Now listen, for example, you have a Japanese woman here who is pregnant. In her blood, she will have IgM against only those infections which she has recent infection. But IgG antibodies are present in body for long time. So when Japanese woman is pregnant, in her blood she has IgG antibodies against the infection which she had 10 years back or 5 years back or 2 years back. So actually everyone has lot of IgG according to the exposure to the antigens in the past. So whenever you get a new antigen, you make IgM for recent use and IgG for long-term use. Again, this is very important. Whenever you get any infection, right, immune system make IgM for recent use and IgG for long-term use. So when any infection come again and again, the IgG keep on getting boosted. Is that right? So what really happens that if mother give, let's suppose theoretically, if mother give IgM to the baby, then baby will get IgM only against those antigens which mother received during her pregnancy. But if mother is giving IgG to the baby, it means mother is giving all those protective manners which she developed against all the antigens which she was exposed in her life. Is that clear? So what is more beneficial to the baby, to receive IgM or IgG? IgG. That is why placenta has receptors for IgG. So maternal circulation, IgG bind with the placenta, and then through pinocytosis process, IgG is taken from maternal circulation to the fetal circulation and gifted to the baby.
So IgM is not.
Yeah, IgM is not transported significantly from maternal circulation to the baby circulation. And once baby is born and mother nurses the baby, feeds the baby, then through the milk IgA goes to the baby. So newborn baby is receiving IgA through the breast milk and already have received IgG from maternal circulation. This IgG from maternal circulation stays in baby's blood for about 4-5 months. That is why many babies have a high risk of infections after that time. Right? Any more question? Yes.
What if the mother is Rh positive and the baby's Rh negative?
Okay, she has come up with a question. If mother is Rh positive and baby is Rh negative, what will happen? Nothing will happen, but baby, I hope, should be born. If mother is Rh positive, will she make anti-Rh antibodies?
No.
No. And secondly, if baby has no Rh antigen, do you think baby has any type of risk from anywhere? No.
So nothing ever happens?
Nothing will happen.
Okay.
This is a special condition in which Rh negative mothers are giving birth to Rh positive baby. So the risk is that if it is first delivery, the mother immune system is stimulated to make anti-Rh. And if she has previous deliveries also, then maybe this baby is born, if mother had already developed anti-Rh antibodies, then this baby may be suffering. But if mother is Rh negative and baby is Rh negative, do you think there will be any problem? Not at all.
So they give one time and then they never give it again?
No, they just give, when Rh negative mother gives birth to Rh positive baby, they give injection once after the, delivery of the baby. Then they don't give it. Yeah. Right? Any question? No? Class dismiss.